Hang out on air.
We're
is
good?
Live. Yeah.
Are you recording audio too?
Yeah. I'm recording audio here and here.
You can get these made for
Hello,
audio.
everybody.
Yeah, I think I'm gonna have to do this.
Apparently we are now live
You okay?
streaming, so we can get going. It's very exciting to be here. Hello and welcome to DNA Day. My name is Dr. Catherine Dukars, and I'm honored to be your host and moderator for the panel discussion today on personalized medicine. This is the 6th year that Canada has celebrated DNA Day. This annual event is developed to help young Canadians explore the world of genetics and connect you with the science community. It's also created to raise awareness about DNA and the impact that genetics has on our world. So why do we celebrate DNA Day in April? And I won't ask you to answer. For two reasons. April 1953 was the year that James Watson and Francis Crick published their paper about the structure of DNA, the double helix. And it's also in April in 2003 that the Human Genome Project was completed. So Let's Talk Science and Genome Alberta are the two organizations that have made this day possible. Let's Talk Science is a national education and outreach organization that engages young Canadians in science, technology, engineering, and math, and through programs designed to make these subjects interesting and relevant for your future studies. Genome Alberta is a not-for-profit organization that initiates, funds, and manages genomics research in partnership in Alberta. I'd also like to say a special thank you to Genome Canada, Genome Atlantic, Genome BC, Ontario Genomics, Genome Prairie, and the Ontario Science Centre, the Government of Canada, and the University of Guelph for their support for DNA Day. Today, our panel of experts gathered here will discuss personalized medicine. We're each going to do a quick overview about our research and our work in genomics, and then answer your genetic questions. If you'd like to submit a question to today's chat, for those of you online, please go to Let's Talk DNA CA and fill in the form, or post a question to Twitter using the hashtag DNADayCDN. Before we get started, I'm going to introduce our panel. As I mentioned, my name is Dr. Catherine Dukars, and I'm the director of the Ontario Personalized Medicine Network, and also Senior Manager of Business Development and Research at Ontario Genomics. Our panel today includes, first on my right, Dr. Mark Hume, who is co-founder and CEO of a new company called DNA Stack, and he's going to tell you a bit about that today. Nassim Bonfaret, Master of Science, is a genetic counselor and coordinator of the Genome Clinic, the Molecular Genetics Laboratory, the Center for Genetic Medicine at the Hospital for Sick Children, or Sick Kids Hospital. And last but not least, we have Dr. Arun Tiwari, a research fellow at the Center for Addiction and Mental Health, or CAMI, which is made of it. So I'm going to just start off with a brief introduction to personalized medicine by showing you part of a video that we prepared that introduces personalized medicine. So if I could have the video up, we're going to stop it halfway through, but you can see it on our website if you want to see the whole thing.
What is personalized medicine? It's when medical treatment is tailored specifically to the individual characteristics of each patient to ensure the right medicine is delivered to the right person at the right dose at the right time. And while it's the way of the future, it's an incredible opportunity for us to improve medical care in our province right now. Let's look at how personalized medicine works. In today's world, if a doctor had 4 patients with acute debilitating depression, they do a patient history for each and then prescribe one of the most current antidepressants, with varying results, ranging from total effectiveness to very little effectiveness at all. Personalized medicine offers an entirely different future. The doctor would have each patient undergo a complete genomic analysis and look at their history, environment, and a host of other factors. This data would be fed into a computer where it would be analyzed against billions of bits of information from other sources. And voila, the doctor would have specific recommendations for each patient, including the proper drug regimen and dosage. Here's another case. A doctor is treating 20 patients with rheumatoid arthritis. In our current system, with limited information, he would prescribe high doses of naproxen to all. After 6 months, it's possible that only 4 respond well. The rest would be given initial doses of another drug, methotrexate. Let's say only 4 of the remaining 16 responded well, 8 did not, and 4 got sick from the drug. All entirely possible. Well, the non-responders would then be prescribed either Remicade or Enbrel to the tune of $35,000 a year per person. And still, only 50% would respond. Personalized medicine would be far more precise and efficient. At the beginning, the doctor would look at genome sequencing and a multitude of other factors for each patient that would show who would respond well to naproxen versus methotrexate versus Remicade versus other yet to be discovered drugs, with trial and error virtually eliminated. There would be cost savings because patients wouldn't be given Remicade in instances where we knew conclusively that it would not work. And there would be better medicine with more efficient treatment and less visits to the hospital. Yes, personalized medicine would have a tremendous impact on our province, shifting our focus from reactive to proactive. And with planning, it'll improve healthcare, reduce the incidence of disease, and control healthcare costs.
So I hope that helped give you a little insight into what personalized medicine is. Those 2 examples show you how we can use your genetic information to identify the best drug that will work for you, avoid side effects of the drugs that don't work, and speed up the process of finding the right drug for you. And that is one way that personalized medicine is changing our healthcare. I'd like now to pass the podium over to Mark. Just as a way of introduction, a little thought experiment, just like landing on the moon without putting a hatch so you could get out and explore and discover what's up there. Or like getting a cell phone with all the songs in the world encoded in it, but no apps to play it. The sequencing the human genome would have been useless without the ability to share and analyze the huge amounts of information that are encoded in our genomes. And so Mark and his new company, DNA Stack, are opening the hatch or developing the apps to access this personalized information in our genomes. And he's going to come up and tell you why and how he's doing this.
Thank you so much. Good morning, everyone. It's great to be here and to tell you a little bit about how we're trying to develop the internet for genomics. So this is me. My name is Mark. I went to high school and graduate school right here in Toronto, and went on to develop a company called DNA Stack. And I also worked for the Global Alliance for Genomics and Health, which is also headquartered in Toronto. And I stayed here because there's a lot of brilliant minds in Toronto, Ontario, and Canada that are working on genomics. It's a really great place to do this kind of work. So this is my best friend, Dan. He has cystic fibrosis, which is a recessive disorder, which means that you have to have 2 copies of the so-called broken gene in order to have cystic fibrosis. Delta F508 is the name of the mutation that he's inherited. It's actually not, it's pretty prevalent actually. 1 in 30 Caucasians have this mutation. But Dan was unfortunate enough to inherit 2 copies, one from his mom, one from his dad, neither of which have this disorder. And incidentally, Dan's actually a twin. He's not an identical twin, and his sister does not have cystic fibrosis. So this disease causes Dan to have sticky mucus, which affects his digestive system, which means that he can eat almost absolutely everything. So this is like a casual breakfast for Dan. But an unfortunate side effect that his lungs are affected as well. And so Dan has undergone 2 double lung transplants in his life. He's a real hero, and he's doing really, really well. So just a couple weeks ago, we celebrated his 14th lungs anniversary, we call it. It's 14 years since he had his first double lung transplant. So in order to treat patients like Dan, it's really important for us to understand the genome. So in the last 10 years, there's undergone a genomic revolution where genome sequencing has become 1 million times cheaper. And that means that we can now sequence a lot of people. So it's expected that there are 2 billion people that will be have their genome sequenced, and it's expected that there will be exabytes of data. So not gigabytes, not petabytes, but exabytes of data, and that's more than the data that's uploaded onto social media sites like YouTube. So you could imagine the kind of amounts and volumes of data that we're having to deal with. And we think that just like YouTube and Facebook have revolutionized the way that we interact with people online, we think that sharing genomic information can have a similar impact for how we can treat people with genetic disease. So really, I stole a screen cap from the video because this really captures what we're trying to do. Sharing is really the essence to identifying and curing patients with genetic disease. Because genomics is a comparative science. The only way that we know how to identify the cause of genetic disease is by pooling data together and understanding those unique genetic markers that are indicative of someone's disease. So what we're trying to do is develop what we find an issue with pooling data together is that people speak different languages in genomics. And in order to construct this pool, we found that we really need to standardize. Sorry, the slides are too far. Sorry, are you able to go back? Sorry? Back, please. Yes, perfect. Okay, so really one of the essential problems is that we really need to construct an internet for genomics. We really can't have Canada not being able to speak to other countries. We need to be able to assemble data into a pool. And so just like the internet, which has the concepts of HTML and HTTP, which you might have heard of, you may not understand that this is happening under the hood, but there are standards that have been developed for the construction of the internet that make it possible for us to share this wealth of information. And also we have incredible tools like Google, which will allow us to search across all the web content that's made available. So in much the same way, what we're trying to do with the Global Alliance for Genomics and Health and at my company is to develop an internet for genomics where genomic data can transfer freely between organizations so that we can make discoveries. And also we need essentially the Google for genomics. We need the ability for researchers to be able to search for genetic mutations in patients that are relevant to their study. So this is something that we have developed, really proud of our team. It's kind of the first incarnation of a search engine, a global search engine for genetic mutations, which allows organizations that are sequencing patients to be able to share with the world the genetic mutations that they've observed. Okay, but with great power comes great responsibility, not to be pointing, but sharing genetic information implies that we're sharing with the world that which makes people fundamentally unique, right? So DNA is your personal ID card, and unlike your internet account, you can't just change your password if someone's discovered it. So it's really, really important that we take the appropriate precautions in terms of privacy and security. It's also really important that we educate and outreach on the importance of DNA and really preach that knowledge is power. If you know your genomic sequence, you can take proactive methods to improve your health. And finally, we need important policies that proactively will protect us from misuse of genomic information. So my opinion is that precision medicine is extremely powerful and will rapidly accelerate with the development of internet for genomics. But there are really important challenges that we need to face along the way, but they're definitely worth it. I think the benefits of sharing genetic information far outweigh the costs of not sharing. So thanks.
Thank you, Mark. And so next up, I'm going to have Nassim. And Nassim works with patients and their families as a genetic counselor who come in for genetic testing and for counseling. Genetic testing raises complex ethical issues. What genes do you test for? What do you find? What do you do if you find something you weren't looking for? Who do you tell about this genetic information? Which members of the family have the right to know? Naseem and her genetic counselor colleagues deal with these questions every day.
Thank you. I work for the Center for Genetic Medicine at SickKids, and I'm just going to talk to you a little bit about the clinical impact of genomic medicine or personalized medicine. And I'll show you a case, an example, and hopefully that'll make things sink in a bit more. Okay, so just to review, you probably know this, but we have billions of cells in our bodies. In each cell, we have a library of genetic information. And you can think of this genetic information as a kind of a storybook. So we have about 20,000 stories, and they're packaged into these books called chromosomes. So when we talk, sorry, can you go back? Okay. So the DNA, or the genetic information, is coded in 4 letters, C, A, G, and T. And if you look at any location in the genome, people will have different letters at that location. So if you look, sorry, why is this going forward? Can you go back to the second slide, please? So if you look at that picture with the 3 people, so at the 4th letter, someone might have an A, another person might have a G, another person might have a T, and that's just normal. That's part of being a human. But when we talk about genetic and genomic testing, essentially what we're doing is we are taking a person who has a genetic condition, and we are looking at their genes, and we're trying to figure out, is there a mistake in the genes that would explain what they have? So if I have a child with a heartbeat problem, what we're trying to do is we're trying to look back at one of their 20,000 genes and figure out, is there a mistake in one of those genes that explains the heart problem? So, sorry, I'm bad at this. Can you go one back, please? There, thank you. So up until about 2000, the year 2000, when we talked about genetic testing, we were looking at 1 gene, 2 genes, 3 genes, maybe a handful of genes for each person. And that allowed us to provide an explanation or a diagnosis in about 10 to 15% of cases. So when you say 10 to 15% diagnosis rate to a genetics person, that's great. To any other person, that sounds pretty horrible. Since the year 2000, there has been a revolution in the technology that allows us to look at most or all the genes at one time. So that cuts down on the cost of genetic testing and the time it takes to do a genetic test. So since then, our diagnosis rate has gone up to about 30 to 50%, which is a huge jump considering where we were to begin with, but we're still not anywhere near perfect. So when we talk about looking at all the genes or most of the genes at one time, that's what's called an exome or a genome test. So you probably have heard those terms. So when we say a genome, that's what we mean. We mean looking at all the genes in one go. That doesn't mean that we can interpret all the genes, though, or interpret what we find in all the genes. Okay. So there are a lot of misconceptions and unrealistic expectations associated with this kind of testing. So you would think if we look at all the genes, why can't we diagnose everyone who has a genetic condition? And some of that has to do with limitations of the technology. Most of it has to do with our limitations in terms of analyzing and making sense of what we find. So we have about 20,000 genes, each of us. We have 2 copies, 1 copy from mom, 1 copy from dad. Out of these 20,000 genes, we only know what about 3,000 to 5,000 of them do. So the rest of the genome is a sort of a mystery. So we're not sure what those genes do. We're not sure what changes in those genes would do. And a big part of sharing data and doing research is really trying to hone in on, can we make sense of these genes if more than one researcher in the world is looking at them and sharing the data that they find in those genes? Okay. This pie chart represents a genome. So let's say each colored dot represents a change in the genes. So if you look at any person, if you look at you or me, and we do a genome sequence, we will find millions of changes in our genes. We call these variants, and 99.9% of these changes are what we call benign. So they're normal, they're just part of being a human. That's why I might have curly hair, you might have straight hair, one of us will have blue eyes, another one will have brown eyes, so they don't really affect our health. But there is a portion of these genes where changes will cause disease. So let's think about a child who has a heart problem, for example. If I see this child and we do a genome sequence, we'll find millions of changes in his genome. Most of these changes, if you look at that gray upper portion, most of these variants are in genes where we have no idea what these genes do. So we can't really say anything about them. There will be a small proportion of those changes that will be in heart-related genes, though. So that's promising. That's what we're looking for. Why does he have a heart problem? So those are variants or changes that we would like to follow. Then there's another portion of these variants that are in genes that are important for drug reactions. So is he going to respond well to warfarin, for example? Or if he ever develops cancer, should he be given drug A or drug B? And then there's another section of those changes that are in genes that have nothing to do with the heart, but predict risk of other diseases. So for example, you know some genes are important for heart function, some genes are important for brain function, some genes are important in cancer development. And when you do a genome, essentially you're looking at all of those changes. So you might find something that is related to the heart. You might also find something that has nothing to do with the heart, but is important for that person's health. So this is sort of where the frontier of clinical genetics at this point, where we have all this information and we can tell people about the risk of future disease, but we're struggling with, should we tell? Who should we tell? And what does that mean for the healthcare system and that individual if we tell them, well, in 10 years, you have a high risk of X, Y, or Z? Okay, so here's the case. So we have a 7-year-old boy with a history of that heartbeat problem, and he also had seizures. So we did genome sequencing, and we figured out why he has the heart problem, but we also found that he has a change in a gene called BRCA1. This gene is important in risk of breast and prostate cancer. And usually people don't develop breast and prostate cancer until they're in their 40s, 50s, 60s. So essentially what we're doing is we're predicting risk of future disease for this child. And if you think back to any genetics that you've had, we also inherit our genes from our parents. So if this child has a risk of breast or prostate cancer, what does that mean about his parents? That means that one of them potentially has the same risk. So then the question is, do we tell the child at 7 years of age that when you reach 40s or 50s, you have an increased risk for prostate cancer? Do we test the parents and tell them, well, one of you has a risk of breast cancer, for example? That will be important for the health of that parent. Because let's say his mother has the BRCA mutation, then she would need to get mammography, MRIs, and we could potentially prevent breast cancer in her. But that would mean that the whole family now knows that there is a mutation in this child that would give him an increased risk of cancer 30 years down the road, and his siblings might be at risk, his parents might be at risk. So what does that do to the child? Is he going to be worried for the rest of his life? Is that going to affect the way he plans his life? Is that going to affect the way his parents view him? So these are the issues, some of the ethical issues that people are dealing with this kind of testing. It's great, it's really wonderful that we can give this much information out, but not everybody wants that information. And when I talk to patients, some people want to know everything. Some people don't want to know anything other than, why do I have a heart problem? And some people have more nuanced opinions where they say, well, if it's cancer, I want to know, but if it's risk of Alzheimer's, maybe I don't want to know that. And there's also the issue of insurance discrimination. So if you tell a 7-year-old he has a risk for prostate cancer, and then at 20 he wants to get life insurance, what is that going to look like? He's going to have to tell he has this risk. Does that mean he can get insurance, or does that mean he can't get insurance anymore? So these are some of the issues that are that we're dealing with right now, and there's really no right answer. So as you develop and you go into your careers, these are some of the things you might want to think about if you're interested in genetics. And I'm done. Thank you.
Thank you, Naseem. And while you're sitting there, Naseem has raised a lot of questions. You'll have an opportunity to ask questions after our last speaker, and maybe you want to follow up with getting some answers out of Naseem about how they deal with these issues. Our last speaker, Dr. Tiwari, works at the Center for Addiction and Mental Health, as I mentioned. And before he comes up, I want you guys to have a little thought experiment. Let's say you had your brand new cell phone, the dream, you've been waiting for it for months. Maybe you saved, maybe you whined to your mom, you got one. It's amazing. And then it stops working. It stops waking you up in the morning. Alarm doesn't work. Sends your text to the wrong people. It's getting bad. So you take it in for repair, and the repair shop takes it in that back behind that screen and comes back and they say, well, it's certainly broken. We got 40 options for you for repair. Not sure which one's gonna work for your cell phone. So I like this one, so let's try it. You go away, see if it works. If it doesn't work, come back, we'll try another one. And this goes on, and in the meantime, it's getting worse. Your projects you sent to school aren't going in, your texts you sent to your friend is going to your mother. It's getting really bad. So you wouldn't accept this, right? You'd expect to go in and get an answer and get the right fix for your cell phone. But this is exactly the situation in what we call trial and error prescribing for some medical conditions such as depression, where we don't really know the best medication for each person, but we do know that there are very strong differences in how people respond to different antidepressant medications. And Arun is going to speak to you a little bit more about his work in solving this problem.
Thank you, Kathy. So I work mostly with the mental health, and we're looking at different psychiatric disorders like depression and schizophrenia. So today I will take an example of depression, which you already saw in the video. So what happens in case of depression is most of the time, most of the patients are treated with antidepressant drugs, and more than half of the patients, they do not respond to the medications and or develop side effects. So if you take a theoretical example here, and if you look at the individual, if a person takes a drug, and if you look at the individuals in the blue category, and look at the first and the second block marked there, so the person carries a T or a G variation at this locus. So this person will respond to the medication and and probably the depression symptoms will go away. But there is a case like a person in the yellow category, he might have a C variation at the second position, which might be related to non-response. So the depression symptoms won't go away if he or she takes the same drug as the person in the blue category. Then there's another case where the person is in the pink category and it has a different variant at the first locus. So in this case, the person will develop severe side effects. So my research is focused on predicting which of like if a person takes a particular drug, which person will respond to the medication and which will not respond or develop side effects. So whenever we take any medication, whether it's an antidepressant or an antipsychotic drug, most of the metabolism for these drugs happens in the liver. And the main enzyme that is involved in the metabolism of these drugs are called the cytochrome P450 enzyme, and CYPs for short. And so these enzymes control the level of the drug after the oral administration, how much of the drug would be in your bloodstream. So of these, there are more than 50 different kinds known, and they have different genetic variants in them which affect their activity. So some of them are listed here. I'll talk about two of these that we use in our laboratory. So if you look at this first enzyme, CYP2D6, so it's a very diverse enzyme. When I say diverse, it means it has several different variants. So on the top left, so this gene can have multiple copies, which makes it so that there's too much of enzyme. And the person can break down the drug very fast. And on the extreme right is a case where where a person does not have the active enzyme or the gene is completely absent. So in this case, they will not be able to break down the drug which goes through this particular enzyme. So here the example is of an antidepressant called nortriptyline. So a person who is a poor metabolizer or who is unable to break down the drug, they would require a very small amount of drug for the same therapeutic benefit. Whereas a person who's a fast metabolizer will require a very high amount of the drug for the same amount of response and benefit. So if you give all these individuals the same amount of drug, the person who is not able to break down, they will have a very high level of drug and they might develop side effects. So this is what is also observed clinically. So similarly, there are other enzymes also like CYP19, which again metabolizes more than most of the known antidepressants. Another point here is that all these drugs are metabolized by several different cytochrome P450. So there would be one enzyme which is doing 50% of the metabolism. There's another enzyme which is doing 30% of the metabolism. So we have to take into account the whole genome and look at all the mutations which might be affecting the response to the drug as well as controlling the level of the drugs. So what we have done in our laboratory is we look at over 60 different variants. Besides the cytochrome P450, we look at the receptors in the brain where these drugs act, and we have come up with a very simple interactive report through which we tell the physicians like which drug to use. So in this case, like this is an extreme case where the person can only take the drug in the green bin, which is here is desvenlafaxine, and the primary reason why most of the drug is in the red category is because the person is not able to metabolize the drug, or he or she has a brain receptor which does not respond to the medication effectively. So this is and we provide this report through our pharmacogenetic research clinic at CAMH, and this is available for free of charge for the province of Ontario, and we have provided this information to over 5,000 people in the past 2 years. And this is how we are trying to bring personalized medicine into the treatment of psychiatric disorders. Thank you.
Thank you, Arun. And so that is a peek into how research is changing and going to change very soon the treatment of patients with depression and other psychiatric disorders. And the faster you reach the right cure, the better the patient is, because they don't descend further into deeper depression. And it's a really interesting work, and it's happening right now in our city. So now it's up to you. We are going to open the microphone to questions both from our audience here and from our audience online. And as well, we have some questions that were pre-submitted, and we can probably start with a couple of those while you guys get up the nerve to come to the microphone. I know you want to ask a question, so come on up. We'd love to hear your thinking. And then after that, you guys are going to have some fun. I think you're going to isolate some DNA. It looks like some fun setup there. So if we could have some questions up, we can start. I remember one of them. A couple of the questioners submitted a question about genetically modified babies and how realistic this was. Was it going to happen soon? And what are our thoughts about that? And this is a question I'm going to turn over to Nassim.
So this is actually one of my favorite questions. A lot of people ask this question. Designer babies are not happening now. I think with even a very optimistic estimate, we are many, many years away from being able to design a baby. However, we're a little bit closer than we were, let's say, 5 years ago. So I assume by designer babies, people don't really mean eliminating a disease change or a mutation in a fetus, because that's more for a medical reason. But if we're talking about choosing traits for a child, like hair color, height, those sorts of things, technically it is probably going to be possible in the next 10, 20 years, because there are new technological advances like this CRISPR technology, where you can cut out a piece of DNA and replace it with something else, and that will give you the trait that you're looking for. However, there is a huge resistance in the genetics community and the ethics community to this kind of use of technology in this way, because really, despite what people may think, genetics professionals are not interested in eugenics. We're really not in the business of creating the perfect race or anything like that. So technically, it is probably possible in the next 10 to 20 years. Ethically, I don't think it will happen, and that's probably for the best. Being able to correct disease traits, for example, so for example, this cystic fibrosis trait, that's probably possible. Or if someone has a muscle disease where they can't walk, it's probably going to be possible for us to replace that gene with one that's working. And I don't think many people have a problem with that, although it hasn't happened yet, so we're still in the thinking phase.
Thank you. And I see we have a question on the floor.
Hi, my name is Tabrish, and my question is, do you guys see any issues or concerns with the privacy of like uploading everyone's genomes into a database and storing it?
Good question, and I'm going to look at Mark for the answer.
Yes, for sure, and definitely, you're not the first person to bring this up. It's a very common question that we have, and it's super complex. And so we have to consider the technical aspects of securing data, and also the ethical aspects, as well as the regional concerns. So different countries have different legislations as well, and so it requires us to have a conversation globally about how we construct systems like this. But it's very clear that we need to do this on a global scale. We can't have our own system in Toronto and a system in Boston, for example. And I think that just like the internet is constructed so that no one organization owns the data is really, really important. So just like the internet, you're able to boot your own website up on your home computer. That's what we're trying to achieve with genomics, so that at least there's some sense of ownership in terms of whoever owns the data is able to make that available online. So I don't think there's going to be one database, but a set of databases that are connected gives a little bit more control.
I'm gonna ask a follow-up question from that. So what about the fact that DNA is so unique to each individual that you can identify an individual from their DNA that might be on the internet? And how do you protect people from the misuse of that information?
That's another good question. It's actually, so the proof of concept that I showed, that website, was a search engine for genetic information. Someone conducted a study where they showed that statistically, if you ask the question enough times, you're able to identify, yes, in fact, Kathy is part of this particular database. And so this is an issue of identification. Even though we don't say publicly that you're listed in the database, we can confirm using some tricks whether you're in there. So it's a really, really important question. The way that we're solving this is, I think, mostly through policy. So we're right now trying to implement some hurdles for people to go through in order to confirm that they are who they say they are, they do have a good purpose, and to make sure that their purpose for accessing the data corresponds to the consent that patients have given for their data to be used. So when a patient gives up their data for research purposes, for example, it shouldn't be used by a for-profit company. And so we're trying to implement these kind of enforcements for access to the data, but that's a great question.
Any other questions? Somebody's coming up to the microphone.
Sorry, what do you think of companies like 23andMe that are providing information to oneself? And how do you think that we can manage responsibly how we deal with that information and the possible disease risks that we might have?
Steve?
So I think companies like 23andMe and others have sort of, there are pros and cons to what they're doing. Thank you. So they've kind of opened up the genetics field to everyone. So you can go out there and you can look at your genes and try to figure out, do I have an increased gluten sensitivity or an increased risk for this or that? But I think what's important is to realize this is not medically actionable information. So what they look at is not typically what we would look at in a clinical visit. So they can tell you, well, you have a 1.5 times increased risk of heart disease. Sometimes you can tell that by just looking at your family history. So I think it's important to put that information in context, not read too much into it, but on the other hand, realize that this does empower you to take different options in your life, for example. So if you know you have an increased risk for heart disease, well, you could modify your exercise, you could modify what you eat. I think the danger there is reading too much into the information that you're given. And some people take that as sort of the end-all, be-all, and this is all I need to know. If they don't say I have an increased risk for heart disease, for example, then I'm free and clear and I can do whatever I want. And I think it's just we need to do a lot more education around these kinds of tests so that people know what they're getting and then they can know how to use that test. But otherwise, I mean, people are free to do whatever they want. We don't really, in clinical genetics, we usually call these kinds of tests recreational because they don't really give you medical information. So as long as you realize that, I think it's a good thing.
And I'm going to toss this next question that came online to Arun. The question was asked, I'm sorry, we have one here. Well, that was the question I was going to do. How long will it take to create personalized medications?
So my take on that is like personalized medications are already here. Like at least in case of cancers, we know that there are certain receptors to which the drugs can be targeted. So different types of cancers can be targeted by different drugs. So this is already being implemented in case of cancers. And in case of mental health disorders also, like what I showed you the information, that we are already providing this information to the physicians. Like we do not directly provide this information to the patients because of ethical issues, but we are already giving this information, and this is to the physicians, and this is already being implemented in the clinic. So I believe personalized medications are already here, and the personalization is not in terms of each individual, but it's based on a group of individuals here. So maybe in the future there would be drugs specific for certain traits, but those would be specific to very rare private variations only.
And I'll add a little extra bit to that answer. Drug development pathways are shrinking because of the increase in information about what is actually causing the disease. So you can target the actual genetic change in a tumor, for example, by knowing that information, and far more rapid screening technologies to pick out the best drug or to develop an antibody that's specific for that mutation. We're actually compressing some drug development pathways from the 10 to 15-year process down to, one recent example was only 3 years. So this new personalized information is having a big impact on the efficiency of developing drugs and on the number of drugs that we now need to be specific to all the many complex genomes that are out there. So it's accelerating the process, but it also means that we need more drugs. And we have another question from the floor.
So one of my questions is, what will be the effect on the career of doctors if we just run everything through genome testing? Like, does the career of a doctor become obsolete, or what happens?
You want to? Okay.
That's a very good question. The short answer is we don't know. So part of the challenge is you have all this data, but how do you make sense of all this data? So most of what we find in terms of changes in genomes, we do not know how to interpret. So at this point, you still need clinicians who will look at this patient and say, well, okay, your genes say you have an increased risk for a heart problem, let's say. But you're 40, you don't have a heart problem. This is probably a red herring. So we're still in the stage where we can't interpret what we find in the genes without the eye of a clinician. But we're transitioning more and more to a sort of genotype first kind of test where you see a child and you think, okay, he has X, Y, and Z. I have no idea what to test for. Let's do his genome and then work back. So that transition is happening, but we're not at a point yet where we don't need an interpreter. And so that's probably where the physicians are going to transition.
And the next question is, what are the ethical issues and concerns behind this type of treatment, and how does Canada deal with ethical questions that arise? And I'm going to get back up here.
If we get away from the ethics of even offering this kind of test to a family or a patient who might not quite understand what's happening and what kind of information will come out of this kind of testing, there is the issue of, we live in a country where the healthcare system is public, so we need to think about costs to the healthcare system. Do we have the ability to do genomes on every kid who walks into a hospital or every adult who has a condition and walks into a hospital? We don't have that ability yet. As the tests are becoming more and more cheaper and cheaper, we're probably going to get to a place where ordering a genome or an exome is going to be just as expensive as ordering any other genetic test. So the idea is if we get to a place where a genome costs $1,000, well, then we're in the genomic medicine era. And we're getting close to that. The other issue is sort of this idea of justice in healthcare and equity of access. So if you live in Toronto or Montreal or Vancouver, you have access to tertiary centers, you have access to very specialized care, so you're probably more likely to have access to this kind of testing and treatment than somebody who lives in Nunavut, for example. So we have to think about this idea of how do we make sure that everybody has access to this, the same sort of quality of care and the same kind of testing. And the other issue is we're new, we've jumped into this with both feet, but we really don't know what the downstream effects are of identifying all these risks that we then have to follow up on. So what does that do to a family to know, well, for the next 20 years, I'm going to have to worry about this risk that may or may not happen? How does that affect their psychological health? How does that affect the way they use the healthcare system? So these are questions that are being raised and people are looking at them. Unfortunately, we don't have enough data to say one way or another. So this kind of testing has really been utilized for the past 5 to 10 years. It's been more utilized in the US, but their system is so different that we can't really translate much of the information. So there will probably be answers, but right now we don't quite know.
So I'm going to ask you guys a question. If you could have a genetic test that had medically actionable information in it for diseases that you might experience now or that you might experience in your future, how many of you would want to do that test now? Some keeners? Yeah. And so it's, we're used to knowing about how tall we're probably going to be because we look at our parents, and we're used to knowing about, heart disease because we can look at our family history, but we're not used to knowing about our genetic predisposition for disease. But that is something that we're going to be working on, and we all have to be learning it as a population about the importance of that information and its relevance to our day-to-day lives. Was somebody else coming up there? So the next question is, if the wrong person consumed a tailored medication, what could be the possible effects? And I'm going to point to Arun for that one.
So in this case, like probably the drug might not work. That would be the first answer. Or it depends like how tailored the medication is. Like if it is targeted to certain receptors, then the effect of the drug might not be there. Or sometimes if it has strong effect on those receptors or drug metabolizing enzymes, then there might be other side effects also.
So they might get the side effects, but not the benefits.
Yeah.
Yeah. Question from the floor. Hello.
Hi, my name is Audrey, and I was wondering if when you personalize someone's medication, is there a chance that the medication actually might not work for them?
Arun?
Yes. So because at this moment when we are looking at the genome, we are only looking at a very small part. At least in case of mental health, we do not have enough information about the whole genome and how does it affect your drug response. So we are looking at a limited number of locations in the genome only. So we can predict only to a certain extent whether a person will respond to a medication or not, but there is still some uncertainty in this prediction. So there might be like, although the person is more likely to respond to a medication, but it might be a case that the person doesn't respond because they have additional rare variations that are not covered by the tests that we are offering at this moment. So there could be a case where even if based on our tests, the person should respond to the medication, but they will not probably respond to the medication.
Oh, this question came online. Is it possible to tailor medication so that an individual is no longer allergic to it, for example, penicillin? We had a chat about this before, so I think I'll give you the answer quickly, is that essentially the solution to that is to find other drugs like penicillin that have the same effect. And we do have that. We have many options to penicillin. So that is sort of de facto how we've been, by trial and error, finding alternates to medications that people are having problems with and bad side effects. Question from the floor?
Hi, my name is Amy. I was just wondering, like, you mentioned that there are a lot of different genetic markers that we currently don't know what they're for yet. So how long will it take to actually like look at all the genomic evidence, and what are the current difficulties with finding out that information?
I think I'll turn that to Mark.
Sure. Yeah, great question. I think the answer to that question is how fast that we can get the widest scope of data that we have available to us. We really need to be able to disentangle between what makes us unique, in our hair color and eye color and other features that we have that make us unique from our unique susceptibility to disease, right? So it really is a game of let's assemble large enough cohorts of data so that we can make these determinations. There was a really great effort in synthetic biology, which is basically constructing an organism from scratch. And I think 75% of the genes that they included in that organism, they actually didn't know the function of. And so it's kind of surprising that there's a lot of the genome that we still have not a lot of idea about, and it's just for the most part about collecting more information.
I have one other question.
Okay.
If we were able to unravel the complete human genome, what do you think the ramifications would be if we were able to manipulate it?
If we're able to rapidly
manipulate it. Is that possible?
Well, we actually have some pretty cool tools now. So the CRISPR-Cas9 system was mentioned earlier, and that is a very precise way of individually changing genes, and that's just revolutionizing research, making it much faster and much sooner to get to that point where we understand all of the genes together. So we can do that, but there's still understanding how 20,000 different genes work and how they work together, because they all work in closely interconnected pathways, and manipulating one thing, there might be compensatory mechanisms downstream that eliminate any impact from that particular gene. So it's difficult to unravel what each one does. So as Mark says, It's going to take a lot of data.
Hello, I'm Jordan. Current pharmaceutical companies are producing lots of drugs that target like a wide range of people. Once the medicine for specific individuals comes about, will that industry be dominated by companies who can produce like relative to what we have now, small amounts of specific medicine, or will that not come about until the prices for those specific medicines can drop in comparison to medicines that are widely produced for a lot of people?
So I think I'll take that on if you guys don't mind. So it's already changing. So the old model of drug discovery is called the blockbuster model, where you find one drug that you think will be effective on the most people possible and hit the billion, 3 billion market share. But companies realize that we've gotten lucky, but that model no longer works. And over half the drugs in the pipeline now are what you would call targeted medicine or personalized medicine. So this change is already happening. But your question is how specific are we going to get? Are you going to get medication created for each person for them? And the answer is you could, but at what cost? And the new personalized medicines that are coming out with a bigger price tag because the drug companies are spending almost as much money to develop them, but for far fewer people. So they're coming up with price tags like the new hepatitis C treatment, which is revolutionary because it cures, but it costs $60,000. But you have to balance that cost against the cost of somebody continuing on in their life with hepatitis C and the impact that has on their life and the impact that has on the healthcare system. So $60,000 might not be so expensive when you add that all up. But can we each afford to have a personalized drug made for us? You know, until you get, probably not if you're talking about cancer drugs and other drugs. We'll try and make many, many, many more drugs that you can pick from that will be the best for you. You come into specific drugs when you start talking about maybe stem cell therapies, and that's in its infancy, and that's another part of the personalized medicine story. So yes, coming. Okay, we have time for one more question.
Hi, my name is Jessica. I was just wondering, what's your opinion on the laws and regulations regarding genetics? Because I know with the CRISPR, there was some genetic research done in other countries because it was legal, whereas others it was illegal. So what's your opinion on that? Or
the laws about doing genetic research.
Yeah.
I think I'll turn that one to Nassim.
So you're absolutely right. Some countries are much more permissive about the kinds of research that can be done and the types of samples people can use. Unfortunately, there's really nothing we can do about the laws of a country. What tends to happen now is people focus on creating specific work groups in their own field. So there's ethics work groups, genetics work groups who come up with these guidelines about what should be done and on what sample and with what kind of procedure. So for a researcher to be in good standing, they probably should stick to those guidelines. However, they also have to think about funding models and in science, you publish or perish. So the first one to get to that animal model wins. So there's a sort of tension between the guidelines that come out of different societies in terms of what should be done and the need to get this moving forward. I think right now, especially with the CRISPR technology, there's a huge push to get some kind of guidelines out there so that this doesn't get out of hand before. But there is no guarantee that we can enforce that. Unfortunately, that's a good question.
So unfortunately, that's it for our question period. I'd like to thank everyone who joined us for today's conversation, and thank you for your excellent questions, including the people who submitted them online. We hope DNA Day helps inspire you and maintain your interest in science, technology, engineering, and math. And these topics are all around you, and they're playing an increasingly important role in your life and in your career choices. So if you're interested in genetics, I'd hope hope you'll explore the jobs available in that field. There's something for everyone, whether you come from a perspective of somebody in the arts or somebody in the science. There's many ways to be involved in the genetics evolution that's happening now and will change how you change your life. Today's discussion was made possible by Let's Talk Science and Genome Alberta, and I'd like to thank them. These organizations offer other opportunities for you to explore the world of science, technology, and innovation and connect you with the science communities. So check out their website and follow them on Twitter and Facebook to learn more. Support for DNA also came from Genome Canada, Genome Atlantic, Genome BC, Ontario Genomics, Genome Prairie, the Genome Family, and the Ontario Science Centre, the Government of Canada, and the University of Guelph. So if you'd like to continue learning about DNA, jump onto explorecuriosity.org and search for articles and videos about genetics, So thank you again for joining us for DNA Day, and we hope you'll join us next year for more. Thanks very much.